What if you could cure cancer with just a few shots? I'm Caitlin Savoy, and this is the story of exo-siRNAi. Before we get into the details, let's unpack that title. We have 4 parts: exo, si, RNA, and RNAi. RNA is a nucleic acid used by organisms as a template to make proteins, the workers of the cell. RNAi stands for RNA interference, a mechanism using double-stranded RNA to directly control protein production based on similarities to target RNA. Many types of RNA can be used in RNAi, but here, the Si tells us that small interfering RNAs are being used. These are essentially chopped up pieces of longer strands of dsRNA, but more on that later. siRNA can be endogenous, coming from inside a cell, or exogenous, coming from outside a cell. The exo lets us know that the siRNA here is exogenous. To put it all back together, exo-siRNAi is a similarity-based method of controlling protein production using siRNA that comes from outside the cell. But how does it work? There are 4 basic steps. dsRNA enters the cell, is cut into siRNA, and loaded into some molecular scissors, which then slice the target RNA, preventing a specific protein from being made. Let's zoom in a little more. The specifics of RNAi differ by species, so we'll use fruit flies as a model. There are many types of dsRNA, specific to various viruses and organisms, each with its own way of entering the cell. Once in the cell, it's bound to a protein called dicer, which, cleverly enough, dices the dsRNA into shorter double-stranded pieces of siRNA that have a small overhang on both sides to identify themselves. Now ready for loading, another dicer protein and a protein called R2D2, no, not that R2D2, bind to opposite ends of the siRNA with the help of a third protein, TAF11. A 4th protein, Argonaute2, enters the scene and takes the siRNA from dicer2 and R2D2. Only one strand of the siRNA will be needed, the guide strand, so AGO2 cuts away the other, known as the passenger strand, to be broken down in the cell. Additional proteins bind to the AGO2 siRNA couple to form the RNA-induced silencing complex, or RISC, the previously mentioned molecular scissors. Fully prepared, the guide siRNA leads RISC to the target RNA, which is nearly complementary to the guide in structure. The siRNA binds to the target and AGO2 slices it, making it impossible to use as a template for protein production. This way, a protein of interest is prevented from being made. But why is that important? Many organisms like fruit flies use exo-siRNAi as a defense against viruses. There's evidence for this in mammals too, but not enough to say for certain just yet. Outside of normal biological functions, the process can be used to study gene function and engineer better foods, such as by removing allergens. To do this, dsRNA is delivered to cells via shots, food, or surface contact. Efficient delivery and breakdown remain obstacles for the moment, but in the future, it may even be used as a species-specific pesticide or treatment for disease. With a little more research, curing cancer may be as easy as getting just a few shots. Thanks for watching.